बताइए। Good morning, everyone. It is my privilege to welcome all the distinguished guests, respected scientists, research scholars, students from various colleges, and all the participants to the second edition of Innovations in the Medicine Symposium. May I now request all the dignitaries to kindly take their seats on the dais. Dr. Radha Rangarajan, Dr. Swami Subramaniam. 1 year ago, the symposium was envisioned and initiated by our respected director, Dr. Radha Rangarajan, with the objective of showcasing how years of fundamental research are translated into life saving medicines. Today, we continue that vision by celebrating another year of scientific innovation and translational research. This year, we will follow the journey of two drugs from their discovery and development through clinical trials and finally to the FDA approval. We are deeply honored by the presence of our chief guest, Dr. Swami Subramaniam, Director Ignite Life Science Foundation, and we extend a very warm welcome to him. May I now request our respected director, ma'am, to kindly welcome our chief guest by presenting a bouquet of flowers. May I now invite our respected director, Dr. Radha Rangarajan, to deliver the welcome address and formally start the symposium.
Good morning, everyone, and a very, very warm welcome to you for this special event. And of course, a special welcome to our chief guest, Dr. Swami Subramaniam, Director, Ignite Life Science Foundation. Swami has been at the forefront of drug discovery and development through his many roles in Merck, Dr. Reddy's Laboratories, Origin Discovery Technologies, and now at the Ignite Life Sciences Foundation. We're delighted that you so readily accepted our invitation to be here. Swami, thank you. I also want to welcome the students of Hygeia College, Aryavrat College, Amity University, and the Pharmacy Department of Lucknow University. We're very glad that you are you've made this trip out to CDRI, and we hope that this is a good learning experience for all of you. Today's symposium, Innovations in Medicine, as you just heard from Sonam, is the second in the series. And today, new drugs will be showcased. Vepdegestrant, the first PROTAC to ever be approved, and zoliflodacin, the first novel class of antibiotics to come to market in maybe 20 years. These two drugs represent major milestones. One because of a completely new approach to disabling a protein, and the other by virtue of being a niche antibiotic of a novel class. In each instance, the journey to go from lab to market was not a straight line. And what our students have done is to review the literature and the patents and information available publicly to piece together the scientific narrative. They will tell you about the various technological advancements that allowed these compounds to progress and finally reach the market. What makes this symposium really very special is that this is a completely student-led symposium. It's a student-led initiative. The students prepare, practice, seek mentorship, and put it all together by themselves on their own. This is the second student symposium. We do two every year. One, as you know, is the Nobel Symposium, and this is the other one. And so it's really to the students' credit that they put in so much effort to bring this to all of you. And it's about 2 months all put together of work. So the specific students who will present today, Nitika, she is with Professor Shashidhar, and she has been at CDRI since 2024. Shaumya Ranjan Satrusal, he joined CDRI in 2021. He's with Dr. Deepak Datta. Udita Jindal, she joined CDRI in 2021. She's with Dr. Neeraj Jain. Sukanya Verma, she joined CDRI in 2025, so very, very recent. And to her credit, she is part of this, and she's with Dr. Maleshwar Raukuram. Ritu Mandolia, she joined in August 2023, and she's with Dr. Arnav Dasgupta. And Sonam Ranva, she joined in August 2023, and she's with Dr. Amir Nazir. So they're all relatively early in their PhD program, and they've tried their very best. So we're going to listen to them with sort of an understanding of how hard it is to present work that's been done by people who are not necessarily whose work is not always easily accessible. But what they've done is to really create the story from all the information that they could find, and I think you will find it to be extremely interesting. It reminds us that great drugs comes from great science. Of course, this symposium would not have been possible without the enthusiastic participation of our mentors, Dr. Deepak Datta, Dr. Prem Prakash Yadav, and Dr. Arnav Das Gupta. Thank you. It means a lot to us. You took time out of your schedule. You put your heart and soul into it with the students, and it has made all the difference. And I hope that for the students here who are listening in, who are part of the audience, you get a sense of what it's like to do drug discovery from scratch all the way to the patient. And the reason why this kind of a symposium is of value is because it's very difficult to write a textbook on drug discovery and development. Every drug has its own journey, its own path. You bring learnings from the past into whatever project you're working on to make it move faster, better, but it's not a guarantee. The other thing is that drug discovery and development is a highly, highly interdisciplinary process. But as scientists, we are trained to be experts in one thing. And when we do that well, we think our job is done. But in drug discovery and development, your job is not done until you have connected the dots with those to your left and right. And this sort of exercise teaches us the value of that. It shows us what it means to connect across disciplines. And finally, each of us has in our own career or in our ongoing journey seen some part of drug discovery and development. But the story is complete only when everything comes together. And it is hard. It's very, very few people have had the opportunity to be part of a complete story from start to finish. So you can live vicariously by learning about other stories and bring that back into the projects that we're working on in the institute, and also in projects that we will seed in the years to come. So with that, I'm going to hand it back to the students. Thank you very much for being here. We really appreciate your presence. It means something to the students. And just in case you thought this was going to be just another symposium, no, the students threw in a surprise element. So don't leave after the first team is done, because between the two teams, they will engage in some fun activities with all of you. So stay put, and I wish you all a fantastic learning experience today. Thank you.
Thank you, ma'am, for your inspiring words and for your continued vision in creating this platform that celebrates innovation in medicine. Now, I request our dignitaries to kindly take their seats in the audience. We now begin the first scientific session of today's symposium. Our first presentation will take us through the fascinating journey of webdegastrin, tracing its path from discovery through preclinical and clinical development all the way to FDA approval. To deliver this presentation, I would like to invite my colleague Mr. Soumya Ranjan to the podium.
Every era of drug discovery has produced remarkable medicines, but every once in a while, a revolutionary idea emerges. It challenges decades of conventional thinking, and it brings skepticism along with it. But when the idea finally achieves clinical success, this just not delivers a new drug. It brings a paradigm shift in the field of medicine, and reshapes the future of modern medicine. Today, we are going to witness one such paradigm shift. Good morning, everyone. I am Soumyaranjan Sathursal. Along with my colleagues Nitika and Udita, will be taking you through the remarkable journey of the world's first FDA-approved PROTAC, vepdegestrant and estrogen receptor degrader. But before we dive into the science, let's first step into a fiction world. Imagine a city under attack by a supervillain, and the cops are unable to stop the chaos everywhere. When at best the cops can keep him restrained, but even if they manage to lock him away, the villain escapes. Then enters the superhero of the city, the Spider-Man. He quickly, with one wave, catches the villain, and one wave connects to a portal. Then he hurls the villain through the portal, disintegrating him into atoms. Spider-Man is then now free to take down on the next villain, and obviously Spider-Man can do this all day. If we apply this fiction into the field of pharmacology, then you get to know that the supervillain is the undruggable protein, and cops are the classical inhibitors. The inhibitors work via a mechanism what we call as occupancy driven mechanism, because they have to constantly occupy the active site of the protein to inhibit its function. And when the protein gets mutated, the resistance emerges. Now let's meet the protagonist of our story, or I should say the protagonist of the story. That's the PROTAC. PROTAC stands for a proteolysis targeting chimera. It is basically a heterobifunctional molecule which has two binding motifs. One connects to the target protein, another joins to the E3 ligase. E3 ligase is a part of ubiquitin proteasome system. The basic function of the PROTAC is to bring two proteins close together, forming a stable ternary complex as shown in the image. Then the target protein gets degraded. Obviously, Spider-Man doing this all day basically represents a key hallmark of the PROTAC, that they act catalytically. This brings a new paradigm in the field of drug discovery, and it represents event-driven pharmacology. Now let's talk about what basically powers this elegant process. The main engine behind this targeted protein degradation is the ubiquitin proteasome system, or UPS. UPS comprises of a complex of proteins. E1 basically activates the ubiquitin tag, then E2 transfers this ubiquitin tag to the E3 ligase. E3 ligase can then add that ubiquitin tag to the target protein. This polyubiquitinated target protein then can be recognized and degraded by the 26S proteasomal complex, and this elegant quality control system continuously removes any unwanted or the misfolded or the harmful proteins out of the cell. For this significant discovery, three scientists received Nobel Prize in Chemistry in 2004. So this basically laid a foundation for the targeted protein degradation. Now let's visualize how this PROTAC smartly hijacks this ubiquitin proteasome system to carry out its function. So when the PROTAC binds to the target protein here, it brings the E3 ligase close together, forming a stable ternary complex. This E3 ligase then transfers the ubiquitin tag brought by the E2 protein and adds a chain of ubiquitin tags onto that target protein. This polyubiquitinated target protein then gets recognized by the proteasome and it gets degraded, and it is completely removed from its existence. Notice one thing here, that the PROTAC doesn't get used up in this process. It is now free to reinitiate the cycle again, which basically represents the event-driven pharmacology, because the PROTAC molecule can degrade multiple copies of the same target protein. But a significant question arises here. If already the market has several inhibitors, then why there is a need for the PROTAC molecule? Let's revisit the proteome data. Out of the 20,000 protein coding genes by the approved drugs, yet only 10% have been targeted by the approved drugs, leaving the major fraction of 80 to 90% undruggable. because many proteins exist as scaffold proteins, cofactor proteins, and also the protein-protein interactions. So basically, they lack the, defined binding pockets. But PROTACs here in this case doesn't need any defined binding pockets. So it brings the possibility that the undruggable region can be targeted now. But we also need to discuss the relevance of elimination of a complete protein. So let's take an example of estrogen receptor. Estrogen receptor has genomic signaling, and apart from that, it has non-genomic signaling, many scaffold interactions, many cofactor interactions. So when a classical inhibitor is applied, it blocks only the signaling function. Rest many non-canonical function remain intact, leaving the potential to reactivate the protein when the inhibitor is gone. But in contrast, the PROTAC totally eliminates the protein, abolishing all possible functions. So the PROTAC removes all the canonical and the non-canonical functions, basically stating the fact that why to fight a protein when we can completely erase it. Some added advantages of it are enhanced selectivity and the possibility to overcome drug resistance. So we are just not witnessing a new drug here. We are basically talking about a completely new therapeutic modality which have the potential to reshape the modern medicine. Now let's trace the fascinating evolution of PROTACs from a bold idea to its clinical success. So the early 1970s to 1995, it basically brought the discovery of ubiquitin proteasome system, which laid the foundation for this targeted protein degradation. There is a very fascinating story behind the birth of PROTAC concept. The two trailblazers of this field are Dr. Craig M. Cruz, who is a professor of chemistry in Yale University, USA, and Dr. Raymond J. Desaies, who is a professor of biology in Caltech, USA. So they both were invited at a junior faculty awards ceremony to present their work. So after some time, when no one was coming to their posters, they started to talk to each other. And this chemist and East geneticist asked a simple question: what if we combine our expertise? That single conversation basically sparked a transformative idea in the field of modern drug discovery and gave rise to the idea of the birth of PROTAC concept. After 2 years, they published the first peptide-based PROTAC, and for this pioneering work, they received multiple awards like Jacob and Louis Gabe Award in medicine. Thereafter, they switched to all small molecule PROTAC after finding that this peptide-based PROTAC cannot achieve that much clinical success as they have anticipated. So they discovered VHL and CRBN like E3 ligase recruiters, which are small molecules only. Later, Craig M. Cruz, being a professor in chemistry in Yale University, founded a company named Arvinas in 2013, which is completely devoted to the PROTAC development. In the following years, they expanded to undruggable targets, and currently many PROTACs are in clinical trials. But for today's session, we'll talk about vepdegestrant, the first FDA-approved PROTAC. I would like to welcome my colleague Nitika on the stage to explain you the chemical optimization that were required for the PROTAC development. Thank you.
Thank you, Samya Ranjan, for such a unique introduction of PROTAC. So, a very good morning, everyone. My name is Nitika, and I will be explaining chemical journey of vepdegestrant ARV-471 and oral PROTAC. So, on 1st May 2026, FDA approved its first and only oral PROTAC drug, that is vepdegestrant. It is a heterobifunctional protein degrader developed for the treatment of ER-positive, HER2-negative breast cancer. Now, vepdegestrant comprises of three main components that works together. Number one is ER ligand. ER ligand stands for estrogen receptor ligand, a molecule that specifically binds to estrogen receptor alpha and direct that molecule towards the target. Now, which is that molecule that is working here as ER ligand? that is lesofloxacin. Lesofloxacin is a non-steroidal FDA-approved drug that is working as a warhead and target estrogen receptor alpha for the process of degradation. Moving on to the other end of the PROTAC, that is E3 ligase. So as my colleague has already explained what is PROTAC, how E3 ligase work, I'll be explaining the mechanism between Celebron and E3 ligase ligand, which is also known as Celebron ligand. Now, this E3 ligase ligand that here used is lenalidomide derivative. Before moving on to, we need to know what is Celebron. So, Celebron is a protein that is present inside the cell. So, lenalidomide derivative first go and find the Celebron inside the cell and bind to it in lock and key mechanism, and then the process of degradation will be started. Moving on to the third and the most important step of the PROTAC, that is a linker. Linker basically connects ER ligand and Celebron ligand together. But let me tell you one thing, that it is not just a connector. It plays a major role in biological activity, especially in a degradation activity, and also maintain proper distance and orientation between ER ligand and Celebron ligand. Moving on to the types of linker. So there is n number of linker categorized into short linker, long linker, flexible linker, semi-rigid linker, and rigid linker. Now, which linker has been used in webdigastrone and why, that I'll be explaining in next slide. Moving on to the optimization and impact of lesofloxacin. So, this is the structure of lesofloxacin, and I'll be explaining the key binding feature of it. First and the most important key binding feature is this phenolic group. This group is critically involved in hydrogen bonding with the amino acids, and it is essential for high binding affinity towards estrogen receptor alpha. Moving on to the second binding feature, that is this tetrahydronaphthalene core. So, this core is deeply buried into the hydrophobic pockets of estrogen receptor alpha and stabilizes the ligand inside the pocket. In contrast to these two, there is one pyrazole ring, which molecule is not essential for high maintaining the estrogen receptor elements. So now the question arises, where linker can be attached? So linker can be attached in only region which can tolerate the substitution and which is towards the solvent exposed area, and that is this para region. So to understand better, let's look at its ligand binding domain. So this is the crystal structure of lesofloxacin. You can see that all the group of lesofloxacin is deeply buried into estrogen receptor pockets. The only reason that was towards the solvent exposed area is this para region. Now you can see that it is also behaving like an ideal exit vector, and it avoids steric hindrance with estrogen receptor alpha and promote a stable complex. So that's why they have taken para region and selected as to link a connector. Now, moving on to why they have selected lesofloxacin as ER ligand in webdigastrone. The first reason is that it has strong binding affinity towards estrogen receptor alpha and estrogen receptor beta. Second, it has good bioavailability. Third, its chemical structure can easily be modified without impacting the biological studies. And fourth, it has known clinical safeties. So that's why they have taken lesofloxacin as ER ligand. Moving on to the optimization of Celebron ligand. So the first generation Celebron ligand is thalidomide. And by the name of thalidomide, we all are aware of the tragedy that was happened in 1950s. So thalidomide was first synthesized in 1953 and introduced worldwide as a miracle drug in 1957 for pregnant ladies for their morning sickness and anxiety. But these drugs come with a tragedy. That was, it causes birth defect in newborn babies. Scientists were confused. Doctors were in shock. What is happening? What is the reason behind it? And after indeed research, they got to know that there is two enantiomers of thalidomide, R and S. R is for that sedative effect and S is responsible for that teratogenic effect which is causing birth defect. But despite of this side effect, thalidomide is one of the most used Celebron ligand, and also it has strong binding affinity towards the Celebron. So that's why it has used in many PROTACs, because its risk can be manageable in many of the adult patients. So they have tried to use thalidomide in webdigastrone scaffold also, but it was not giving promising results. So they decided to make derivatives of thalidomide, and they made n number of derivatives. And in that process, they got to know that there is two derivatives which is giving better binding affinity towards the Celebron, and that two derivatives were lenalidomide and pomalidomide. You can see in the table also that lenalidomide IC50 value is 1.71, which is comparatively better than pomalidomide and thalidomide. So that's why they decided to take lenalidomide as a Celebron ligand. But after observing this crystal docking structure of lenalidomide, they got to know that there is three positions where linker can be attached. C4, C5 and C6. You can see that. So they have attached linker in each position and noted down its half minimal degradation concentration. And they got to know that lenalidomide DC50 value was 55 nanomolar, C5 lenalidomide DC50 value was 30 nanomolar, and C6 lenalidomide DC50 value was more than 100 nanomolar. So that's why they have taken C5 lenalidomide as a Celebron ligand. Moving on to its key binding features. So first is this glutamide ring. It is mandatory for high binding affinity towards the Celebron, and it is the only ring which is binding to the Celebron. Moving on to phthalimide ring. This part of the ring was not involving in any of the binding to the Celebron, but it is towards the solvent exposed area. Now enantiomer. So they have tried both the enantiomer S as well as R, and they got to know that S enantiomer is giving high binding affinity towards Celebron and good biological activity. Moving on to the optimization of linker. So as I've told you that there is n number of linker, short, long, etc. So they have tried all the linker in this scaffold and noted down its IC50 value to know which linker is giving promising results. So first they have tried short linker, and its IC50 value against estrogen receptor element was 1.94 nanomolar. Long linker IC50 value is 3.14 nanomolar. Flexible linker IC50 value was 9.1 nanomolar, and semi-rigid IC50 value was 1.27 nanomolar. And again semi-rigid linker but with different chemical structure of the linker, IC50 value was 2.88 nanomolar. Then they finally tried rigid linker, and in rigid linker they have got IC50 value 1.12 nanomolar, which is comparatively better than other linker. But the question arises, what was the problem in other linker and why they have selected rigid linker? So the problem in the in short linker was steric hindrance was the problem. In long and flexible linker, the distance and orientation between ER ligand and Celebron ligand was the problem. In semi-rigid linker, the problem was unstable complex. But when they tried rigid linker, there was no steric hindrance. The distance and orientation between ER ligand and Celebron ligand was perfect, and the complex was also stable. So that's why they have taken rigid linker. And in rigid linker, they have taken piperazine linker as a linker. Moving on to the optimization of PROTAC derivative. So by optimizing the ER ligand, Celebron ligand and a linker, the researcher create about 800 PROTAC molecule. After screening, only 9 molecule was selected for further testing against ER positive and HER2 negatives. These are the 9 molecule that has been selected for testing, and among these 9 molecule, one was given best IC50 value, that is 1.1 nanomolar. And then they finalize this compound, this scaffold, as a final optimized scaffold and named this scaffold ARV471. Now, this study shows the essential pharmacophore feature and justify the selection of final optimized scaffold for the further preclinical and clinical analysis. Moving on to the synthesis of vepdegestrant. So first I'll be explaining the Eastern fragment synthesis. So this is the commercially available CBZ isoglutamide ring and isoglutamide group, and it will undergo hydrogenation to remove to form this free amine, and here deprotection occur. Now this free amine will couple with this aldehyde containing fourth intermediate to form a stable carbon and nitrogen bond. After that, this intermediate 5 will undergo acidic treatment and promote intramolecular cyclization to form this glutamide ring, and this is our desirable compound strings from Eastern fragment with 91% yield. Moving on to the Western fragment synthesis. So this is commercially available benzyl protected group. It will undergo hydrogenation to remove this double bond. After removal of double bond, a racemic mixture formed. To form a chiral compound, they reacted this intermediate 8 with diproline, and chiral resolution was done to form this intermediate 9. Now this acetyl group will undergo acidic treatment with sulfuric acid to form this aldehyde, and this is our desirable compound 10 from Western fragment with 80% yield. Now the most important part, that is this 10th and 6th will couple together to form a crude vepdegestrant, and this crude vepdegestrant will recrystallize in DCM, methanol and butanol to form pure vepdegestrant with 90% yield. Now this is the X-ray structure of vepdegestrant. Now moving on to the physicochemical properties of all the oral degraders. So as we all know that PROTAC doesn't follow the rule of 5. So that's why the rule of 5, because of its large molecule and complex structure. So they have studied number of protags, and made a physicochemical properties for all the oral degraders, and name that properties beyond the rule of 5. So first exception is that your molecular weight should be less than or equal to 1000. In case of vepdegastrant, it was 724 Dalton. For log P, that is partition coefficient, should be less than or equal to 7. For webdegastrant, it was 6.8. Your topological polar surface area should be less than or equal to 200. For webdegastrant, it was 96 angstrom. Your hydrogen bond donor should be less than or equal to 5. For webdegastrant, it was 2. Your hydrogen bond acceptor should be less than or equal to 10. For webdegastrant, it was 7. And your rotatable bond should be less than or equal to 10. For webdegastrant, it was 8. So this is the beyond the rule of 5 for all the protag/oral degraders. Now, I would like to call upon stage my colleague Swami Ranjan to explain preclinical and clinical analysis. Thank you.
Hello again. So now we'll be talking about the preclinical validation of this first-in-class oral protag. Let's first understand the disease condition for which vepdegestrant is being developed. ER-positive breast cancer accounts for almost 70% of all breast cancer cases, and there is a high incidence of metastatic progression. Once metastasis progresses, there is a drop in the 5-year survival rate to 30%, and at this stage, it is very difficult to treat the disease. As the name suggests, ER-positive breast cancer rely heavily on the estrogen signaling. Once estrogen binds to the estrogen receptor inside the cell, it brings about the receptor dimerization. Then these dimers translocate inside the nucleus to bind to the estrogen response element that activate the transcription of genes responsible for cell proliferation. Currently, the endocrine therapies that are used in clinics constitute aromatase inhibitors that degrade the estrogen produced inside the cell in the body, and the selective ER modulators, the SIR, basically interferes with this estrogen signaling. One very familiar example is the CDRI shown Saheli or the Saint Croman, which basically works by this selective ER modulator pathway. This is just for our understanding. Saheli is not part of the ER-positive breast cancer therapy. The FDA-approved drugs, tamoxifen and lasofoxifene, as discussed in the chemistry section, are part of the endocrine therapy. But this endocrine therapy poses several limitations like acquired resistance and incomplete ER blockade, and it limits their basically longer therapeutic benefit due to these limitations. So to circumvent these limitations, a next generation therapy came into existence, that is the selective ER degrader. So the FDA approval of fulvestrant in 2002 provided a clinical proof of concept that targeted protein degradation can be used in clinics for the betterment of the patient. But it exists as an intramuscular injection, so it has also its own limitations like incomplete ER degradation, which limits its therapeutic benefit, and acquired resistance obviously eventually occurs. But the main problem is that poor bioavailability and its intramuscular route of administration further poses significant challenges. Let's now discuss how vepdegestrant addresses these limitations. We'll begin by talking about the target binding part. So the team, the scientists who developed this vepdegestrant, they performed a luciferase reporter assay for the estrogen response element, and they found dose-dependent decrease in the estrogen activity, estrogen receptor activity, and found IC50 of 1.1 nanomole. In next in-cell Western immunofluorescence assay, they also found the similar trend of ER degradation with dose-dependent wise, and the DC50 is basically a concentration required for 50% ER degradation, that came around 0.94 nanomole, and with a maximum degradation of 95%. So all these data together concluded that vepdegestrant in fact binds potently to the target estrogen receptor. Now a significant challenge came into their way, that if it is active in the mutation conditions also, because earlier therapies have failed in this mutation condition to produce the therapeutic effect. So in cell line models, they first check the estrogen receptor degradation in the wild type condition, and obviously observed the dose-dependent decrease in the estrogen receptor protein as seen in this Western blot image. Then they replicated the same in the mutation conditions and generated and observed the same degradation trend in the mutation models also. One significant and very frequent clinical mutation is a Y5P7S, which we will also be talking about in next slide. Now they asked whether this ER degradation is selective or not, if at all it is like degrading other protein also. So to find out that, they performed proteomic profiling, and they this revealed that no, ESR1 is the only and most significantly degraded protein amongst all the differentially expressed proteins. In another European safety panel radioligand assay, this further confirmed that vepdegestrant in fact has lowest IC50 for this estrogen receptor. So we can conclude that there are no notable off-target effects that might hamper its clinical use. Now let's correlate this ER degradation to the biological phenotype if at all it produce any result. So in wild type as well as a mutant ER positive breast cancer cell line, the GI50 is basically similar. GI50 is the concentration required to achieve 50% cell growth inhibition. So it came almost similar. Altogether, these data conclude that vepdegestrant is a very potent and selective ER degrader, and it works against both wild type and the mutant ER protein. Now the team wanted to mechanistically validate if at all vepdegestrant follows the actual anticipated PROTAC mechanism or not. Vepdegestrant to ER binding we have already seen in the previous slide. Further, to check for the CRBN target engagement, basically the binding to the E3 ligase. For that, they performed a CRBN probe displacement assay. The principle is simple: lesser the signal, more potent the binding is. So interestingly, they found 17-fold shift in IC50 for CRBN target binding once vepdegestrant is already bound to the estrogen receptor. So it indicates that vepdegestrant and ER interaction greatly enhances the CRBN target engagement. Now they were asked whether vepdegestrant follows the harnesses the proteasome machinery or not to carry out the degradation. For that, they use carfilzomib, a proteasomal inhibitor, which is also developed by the Craig Cruz himself. So after testing with that inhibitor, they found that no, ER is not getting degraded. So they concluded that yes, in fact, vepdegestrant harnesses proteasome machinery only to carry out this estrogen receptor degradation. Then they wanted structural proof for this stable ternary complex formation after checking the target binding to both these proteins. So they performed, they generated cryo-EM structure for the ER, vepdegestrant and E3 ligase complex, and provided the in situ proof for the stable ternary complex formation. Now, a biggest question remains if vepdegestrant is orally bioavailable or not, and also if it achieves sufficient plasma concentration to induce its biological effect. To check that, they performed pharmacokinetic studies in both rat and mice, and you can see that the Tmax came around 1 hour. Tmax is basically the time required to achieve maximum concentration of the drug in the blood plasma. So it came around 1 hour, so it indicates that it is rapidly getting absorbed in the blood. Now, just reaching the bloodstream is not enough. We have to check if it is achieving therapeutic threshold to carry out the degradation or not. We can deduce that from two values here. So the Cmax came around 0.39 microgram per ml. Cmax is the concentration, the maximum concentration achieved in blood plasma after the oral dosing. So it came around 0.39 microgram per ml. If we revisit the ER degradation data, the 95% degradation was coming around almost 30 nanomole. So it corresponds to 0.02 microgram per ml of blood plasma concentration. So in fact, Cmax stays significantly higher than this threshold, further proving that yes, sufficient plasma concentration is achieved with vepdegestrant once oral dosing is initiated to carry out the ER degradation. The T half here is almost 4 hours, but you can see from the graph that the plasma concentration that is required for the ER degradation stays well above this baseline. So it is sufficient. Now a key parameter we can look at the volume of distribution at steady state. If the value is more than 1000 ml per kg, then there is a higher probability that the drug is getting absorbed into the surrounding tissues. It is basically important in the cancer biology studies because we can conclude that vepdegestrant achieves the probability of vepdegestrant to get absorbed into the tumors is high, so that it can degrade the estrogen receptors in the tumors as well. From all these parameters, we can derive the bioavailability. It came around 24% in rat and almost 18% in mice. Although these values might seem very moderate, but for a PROTAC molecule, it is like an encouragement for the PROTAC. Because PROTACs are structurally very complex and due to its large size, there was a skepticism earlier that would it be bioavailable or not, and through oral route only. So we have already seen that it achieves sufficient plasma concentration inside blood. And another fact you can consider that PROTACs work catalytically. So even if a small fraction of PROTACs are reaching the bloodstream, it is sufficient enough to carry out the ER degradation. So this is a significant milestone for the PROTAC development after they have achieved this oral bioavailability. They have also observed bioavailability in non-rodent species, and in dogs it came around 65%, and in cynomolgus monkey it came around 27%. Now after seeing that it achieves sufficient plasma concentration, we can ask the question if it translates to significant anti-tumor activity or not. And in fact, in the mutation models and the therapy-resistant models, where the earlier classical inhibitors failed to produce any therapeutic effect. So for that, they first generated a wild-type ER condition of cell line derived xenograft model. Here, the estrogen receptor is wild-type. You can see that at 3 doses, 3, 10 and 30 mg per kg, there is a significant reduction in the tumor growth inhibition with vepdegestrant. And also, vepdegestrant achieves almost 90% of ER reduction in as a monotherapy, proving the fact that yes, it can work as a monotherapy. Next, they generated a very challenging model. They mutated the ER in the cell lines and they generated a PDX model. So PDX model closely mimics the physiological conditions of patients in an in vivo setting. And here the ER is the mutated one with Y537S mutation. So this mutation is in the ligand binding domain, and due to this mutation, the ER protein stays constitutively active and the estrogen signaling is continuously ongoing without any supplement of estrogen receptor. In these particular conditions, the classical inhibitor failed to produce any therapeutic effect. So in this condition also, we can see that vepdegestrant achieved almost very potent tumor growth inhibition, and we can see that compared to the fulvestrant, which is a classical ER degrader, it achieved more ER degradation at lower doses compared to the 200 mg per kg of fulvestrant dose. CDK4/6 inhibitors are currently a standard of care treatment, and FDA approved inhibitors are the palbociclib and abemaciclib because they block the cell cycle progression of any cancer cell. But eventually resistance arises and these inhibitors fail to produce any effect. So they generated a further therapeutically challenging model where there is palbociclib resistance and also ER is mutated in this PDX model. This model also and this study also showed that vepdegestrant is well active at 10 mg per kg, producing almost 100% tumor growth inhibition. These data altogether states the fact that vepdegestrant have the capability to defy ESR1 mutation and any kind of therapy challenge. But all these data is insufficient without any toxicology parameters to progress into the clinic. So we know that there are no notable off-target effects and there is no hERG liability up to 30 micromole, and they have also observed no cardiovascular impacts on dogs at up to 400 mg per kg. They also performed a separate safety pharmacology study, a GLP compliant toxicology study in both mice and rats, and they found that there are no observed adverse effect level up to 100 mg per kg in rats and up to 90 mg per kg in dogs. So this basically corresponds to a 20-fold safety margin over the effective dose. After establishing all this potent cancer activity and potent anti-cancer activity and the oral bioavailability and the safety margin, they finally have the proof to advance into the clinic. One thing I want to highlight here is that you already have seen that drug discovery is not a single person's job. It basically amalgamates expertise from medicinal chemistry, pharmacokinetics, pharmacodynamics, disease biology, structural biology, and toxicology studies to finally generate a candidate that is clinic worthy. With this, I would like to welcome my colleague Udita to brief you over the clinical journey. Thank you, everyone.
Good morning, everyone. My name is Udita Jindal, and after cruising through the chemical and preclinical journey of Epticastan, now let's dive deeper into the clinical journey of Epticastan. This is a very significant study because this is first successful in human clinical trial for protein degraders. In this presentation, I will take you through the phases of clinical trial, pivotal phase 3 study, and the future of degrader therapeutics. Before moving on to Epticastan, let's just quickly revisit the objective of phases of clinical trial. Phase 1 determine the safety, tolerability, pharmacokinetics of the experimental drug. In oncology, patients are enrolled instead of healthy volunteers. Phase 2 answer how the effective our drug is. Phase 3 is the comparison of our experimental drug with the standard of care. Outcomes move to regulatory approval. And phase 4 is post-approval market survey. Now coming on to phase 1, Veritac was the name given to the study. Before the initiation of the Veritac, there was a considerable amount of skepticism among the researchers that whether or not this big molecule be orally bioavailable, will it be able to regress the tumor and to provide any clinical outcomes? So to answer this, Veritac was initiated. It's an open label dose escalation trial sponsored by Arvinas in collaboration with Pfizer. ER positive, HER2 negative, advanced metastatic breast cancer patients were enrolled, and the primary object was to determine safety and tolerability and to recommend phase 2 doses. Total 83 patients were enrolled in this trial. The trial was initiated in 2019 and completed by 2022. Dose ranging from 30 mg to 700 mg were given as multiple ascending dose, and the researchers have identified that there was no dose limiting toxicity that was reported. The drug was safer and favorable and tolerable across all the doses, and once oral dose was further used for optimization. Now coming on to the pharmacokinetic parameters on day 15, the table shows two important parameters, that is area under concentration time curve and Cmax, that is the maximum concentration of drug in the plasma. We can see with the trend that there is an increase in the exposure of the dose with increase in the concentration. That is a very important character for optimizing drug. The researchers have also found that there is decrease in the amount of circulating tumor DNA up to 99% after one cycle in all dose group. So based on the PK exposure profile, safety and preliminary data, phase 1 identified 200 mg and 500 mg once a daily oral dose as a recommended dose for phase 2 trial. After determining the safety and tolerability, the researchers want to ask how clinically efficient their drug is. So to answer this, Veritac phase 2 was initiated. It was an open label dose expansion trial where 71 heavily pretreated patients prior with endocrine therapy were enrolled. The treatment arms were patient were randomized into 1:1 ratio, given 200 mg and 500 mg once an oral daily dose. And the aim of the trial was to compare the efficacy and safety of phase 1 recommend 2 doses, 200 mg and 500 mg, and to determine recommend dose for phase 3. The primary endpoints were first the clinical benefit rate, which is the sum of complete response, partial response, and stable disease lasting at least for 24 weeks. When we saw whole population together, the CBR is 38%. But when the researchers stratified their population into ASR1 mutant and wild type, they have seen up to or more than 2.5-fold increase in the CBR rates of the ASR1 mutant patient, signifying that vepdegestrant is more potent for ASR1 mutant patient. We can depict from this fact that these mutant tumors are heavily dependent on ER signaling pathways for their survival. So degrading these mutants will result into more clinical benefit rate. When the clinical benefit rate among the two doses separately were analyzed, the comparison was moderate, but statistically it was insignificant. Now coming on to the second parameter, that is progression-free survival. For all patients at both the doses, the median progression-free survival was 3.71, which increased to 5.71 in the mutant patient. When compared separately with 200 mg doses, the whole patients were 3.5 months, like MFS was 3.5 months, and it has increased to 5.5 months in ASR mutant patient. When we compare these two data together, we can say that the maximum therapeutic benefit has reached at the lower dose itself, and classical example of pharmacological plateau. The treatment-related adverse effects were primarily of grade 1 and grade 2 in both the doses. So after determining the progression-free survival, pharmacokinetics, and clinical benefit rate and safety data, phase 2 recommended 200 mg once daily oral dose for phase 3 clinical trial. After determining the efficacy, safety, and tolerability of vepdegestrant, the clinical wanted to know whether or not this drug is better than the standard of care. So to answer that, VERITAC-2 phase 3 clinical trial initiated. It's an open label, multinational, randomized trial initiated in 2023 and completed by 2025 across 27 countries, more than 250 centers. 8 centers were present in India also, including TMH Mumbai and RGCI Delhi. The aim of the trial was to evaluate vepdegestrant in comparison with fulvestrant, which is an FDA-approved selective ER degrader. And the outcome of this study was published in NEJM in May 2025. 624 patients were enrolled in this trial, and the inclusion criteria was ER positive, HER2 negative, metastatic breast cancer patient who were given prior endocrine therapy and disease has progressed after that endocrine therapy. So vepdegestrant is given as second line of therapy, not as the first line of therapy. The study scheme is patients were randomized into 1:1 ratio. Treatment cycle of 28 days were given of vepdegestrant orally 200 mg once a daily dose and fulvestrant as an intramuscular injection of 500 mg on day 1, 15 and 29 on the first cycle and then in the subsequent cycle. So here comes the most important slide of this study, the result of the phase 3 clinical trial. It also answers the question that all these chemical optimization and preclinical validation delivered the anticipated clinical results or not. So this is the Kaplan-Meier survival plot for vepdegestrant and fulvestrant treated patient. We can appreciate the fact that these two lines separated very early on, like around 2 months, and remain separated throughout the treatment. Around 30% patients who were treated with vepdegestrant were alive and progression-free even after 18 months of treatment. The progression median free survival has significantly increased from 2.1 months in fulvestrant to 5 months in vepdegestrant. The hazard ratio is 0.5, that is 43% in the reduction of the hazard of disease progression or death with vepdegestrant. The adverse effects were primarily of grade 1 and grade 2, fatigue or neutropenia. After the completion of this trial, Venus has announced FDA acceptance for new drug application of vepdegestrant in the treatment of ESR mutant, ER positive, and HER2 negative advanced metastatic breast cancer patients. This is the developmental timeline for Vepano. This timeline marks the milestone in the field of protein degraders, from announcing their first clinical benefit results in 2021 to getting FDA fast track approval in 2024 to submitting and providing acceptance of new drug application in 2025. On 1st May 2026, FDA has approved vepdegestrant for ESR1 mutant, ER positive, and HER2 negative breast cancer. Although vepdegestrant have been a breakthrough in the field of target therapy, but there are some limitations and unknowns that needs to be answered. First is long-term safety and adverse effect that are still unknown. The overall survival benefit data is still immature. If there is any resistance, that needs to be characterized. The combination strategies need validation, and the impact of vepdegestrant in chemo-naive patients is yet to be explored. Till now, the researchers had administered vepdegestrant as a monotherapy, but as the resistance appear, the tumor cells drives this mechanism to multiple signaling pathway. So combination therapy is a better therapeutic approach. So here are the list of all the combination trials that are active with the combination of vepdegestrant. I would like to draw your attention on this particular trial, which has just announced by Pfizer. Vepdegestrant in collaboration with atirmociclib, which is a selective CDK4 inhibitor. Both the drug will be used as a first-line combination for previously untreated or chemo-naive ER positive and HER2 negative metastatic breast cancer patient. As PROTACs are advancing in the clinical stages, one question typically arises that how to increase the specificity of these PROTACs. So to answer this question, next generation of PROTAC technology came as a tissue-specific E3 ligases. In current or traditional PROTAC settings, cereblon is used as an E3 ligand, but these cereblon may present in the normal cell also, so they have chances of potential of target degradation. While in the next generation of PROTAC, that E3 ligase will be tissue-specific, so there will be high selectivity, better efficacy, and safety. These are the example of some tissue-enriched E3 ligases. So the key takeaway is the first generation of PROTAC told us how to degrade the protein. The second generation will tell where to degrade these protein. So PROTAC has reformed the field of targeted therapies, but the field is completely new and evolving. So here in brief, I am discussing the new frontiers of degrader therapeutics. The first is RIPTAC, that is regulated induced proximity targeting chimeras. These are heterobifunctional proteins like PROTACs only, but the target protein are mainly the oncogenic protein, so they have more specificity and less toxicity. The second modality is ASTAC, that is acetylation tagging. Many proteins are not mutated or they are not degraded, but they are functionally suppressed. So these ASTAC will reactivate or transcriptionally activate that protein of interest with the help of lysine acetyl transferases. In the ASTAC, the protein of interest are mainly the tumor suppressing genes. So this is a reverse technology or reverse modulation. The third modality is molecular glue. Molecular glue functions are the same principle as PROTAC, but PROTAC are bifunctional protein, but the PROTAC are larger protein, but molecular glues are single monovalent proteins or molecules that binds to the protein target protein and E3 ligase, and because they are smaller, they provide better oral bioavailability than the PROTACs. The third modality is snipers. Snipers are different from PROTAC in the concept that PROTAC use E3 ligase, cereblon E3 ligase. Sniper uses IAP proteins like CAIP and XIAP as E3 ligases. So in conclusion, I want to say that PROTAC has shifted the paradigm of drug discovery, but that is just a tip of iceberg. The field is continuously evolving, and protein cannot be only inhibited, but they can be degraded, rewired, or modulated as per the therapeutic need to provide better therapeutic outcomes. Thank you.
Now we will take a short break of 10 minutes. After that, we will have an interactive quiz session and there is a special announcement regarding lunch. Today's lunch timing has been slightly revised and will be served in the canteen from 1 p.m. to 2:15 p.m. And before that, we are open for questions right now.
Hello. Hi. Very nice presentation. Also, in context of molecular biology, when they did the, before the clinical trials, if the cancer, is there something known about what happens in the cells of cancer when there is overexpression of D-ubiquitin protein? So how does that cancer cell handles that? When you're doing this, is PROTAC as a whole ubiquitination degradation, but there is a whole antagonistic mechanism. Is there something done on that in context of clinical trial or the patient if they have the overexpression or misregulation of those proteins?
So thank you for the question, sir. So as the PROTAC is constantly developing, yes, it is still a demerit for the PROTAC field. But they have like from the literature what we have understood that PROTAC basically works in a catalytic fashion. So if deubiquitinases are like overexpressed, but PROTACs, as long as it stays inside the body, it can carry out its function and can be able to overcome the this overexpression of deubiquitinases. And obviously, it is still a demerit, and the field is constantly evolving to circumvent that limitation.
Thank you.
Any check has been done on the bone density, because particularly when you are targeting ERs, it will directly help target the bone health.
So, ma'am, they have talked about some of the off-target effects regarding the reproductive health in the ND application, but they have also reported that as long as the treatment period is over, then they have regained the bone health and the like the harmful impacts on the reproductive.
Supplements are given.
Yeah, so once the dosing period is over, then they have regained their reproductive health. So it is like they have not shown any significant clinical adverse effects.
Okay. The other question was about the like the mode. I believe it's oral. Yes. Whether any local ever tried?
Sorry, ma'am.
I believe the dosage was oral,
Yes.
right?
Yes.
So any local inside the tumor, it had been tried?
No, ma'am.
No.
Hello, I'm here, Dipankar here. So thank you very much for the presentation. I just have one query regarding the activity of these compounds. So now compared to the original molecule, which was only the inhibitor. So what was the IC50 of that molecule? Either cell-based or cell-free assay with this one. So my question is that after having PROTAC addition, so is the IC50 increased even including the binding?
You are asking particularly about the ER binding or the
Yes.
So
is it increased or decreased?
It is decreasing.
Decreased. How about the cell-based activity?
It was increasing, sir.
It was increased.
Yes, sir.
So it means that if you use the PROTAC to any inhibitor, that means we could put it to the more efficacy with that one.
Yes, sir.
So any rationalization
Rationalization.
that what is the reason for that? While the inhibition, the decreasing, on the contrary, the activity is increasing.
The reason of that is based on, sir, that binding affinity towards the cerebron and binding affinity towards the estrogen receptor. That is only responsible for the activities.
So while the binding affinity is decreasing, but still the cellular activity is increasing. So how do we connect with this?
Sir, I want to answer because binding affinity here in this case, C referred to the IC50 decreased for the ER binding. So as long as it potently binds to the ER, it can degrade the estrogen receptor in the cells potently. So that basically confirms that although IC50 is lowered, because that's what the medicinal chemist want. So as long as it binds very tightly to the ER protein, then it can recruit the E3 ligase as discussed earlier. So it can degrade the ER inside the cells. And obviously, for the structural modification also, there are lots of modification done on various protags. So first they have tested what modification exactly has contributed to the significant ER binding. So from that, they have selected a particular modification and they checked the anti-cancer effect is there and the binding is still potent. So basically, it is like cooperative in their nature.
And just one again query with this one. If you let us know the name of this drug, how the name has been created there.
Okay. So the name, basically the code name was ARV 471, and strand basically decodes for the ER degraders. And WebDigstrand, like they have suggested the name. Yeah, we can look at it, but I don't know.
So
WebDigstrand.
DEG A strand. Est means estrogen, ant means antagonist, and DEG means degrader.
Okay, thank you, sir. Thank you for the learning lesson. Obviously, it is a perennial process. Thank you.
Yeah. Now we can all go for break.
Please come back after 10 minutes. Okay, 10 minutes. Quiz session is waiting for you. Thank you.
Everyone, please be seated. We're about to start the interactive quiz session. Thank you. नहीं, स्टार्ट करें. Hello everyone. We have arranged an online quiz as per the suggestion of our dear director ma'am. So everybody here is welcome to join the quiz. Here are some sort of guidelines. Everyone please should have an internet connection to scan the QR using your phone's camera or the scanner application. And everyone needs to enter their name correctly because it will be displayed on the leaderboard, and based on that only you will be judged. And time per question is almost 20 to 45 seconds per question. uh, uh, Main point is that this is a time based quiz, so the quicker the response the more points we will be getting. So yeah, everybody can scan the QR. Have everyone joined? Okay. So, so I guess everybody has joined the quiz. So we can start the quiz session. The first question. पता नहीं. Everybody should click on the option and submit. Yeah. After the timer has stopped, it will move to the next question. So I guess the person with the name A needs to identify because we can't identify him or her. You all can see the live score leaderboard also. Who is winning or scoring highest? Just remember, this is time based. The quicker you respond, the more points you will be getting. So you should not be using Google or ChatGPT for the answers. So phone में 20-30 seconds है वो per question. And there is a sweet surprise for the winners. If you can remember, we have talked about this in our presentation. You can see the correct answer on the screen when it is highlighted in the green. वो सब बाद में होता है ना, रिस्पॉन्स के बाद. So that's the end of the quiz session. We can identify the winners, but we have to identify the anonymous person. That's why I requested to enter your name correctly. Okay. Slide down. Okay, sir. Okay. So, as it is evident from the leaderboard, we will be announcing the winner's name, and obviously, anonymous is not getting any reward. I now welcome our director, ma'am, to distribute the surprises. And our chief guest, please do the honor. Swami sir, you can also join on the dais. Please do the honor, sir. So the top scorer is Pranoy. Please come onto the dais. Yeah, you can introduce yourself here, please. जाओ, जाओ. Next, Shruti V. Please come to the dais. Shruti V. Now next, Nikita. For the second one, we are not giving an award because it is anonymous. And next, sorry. Yeah. She is from Dr. Sanyal's lab in biochemistry division. Nikita. She is from Dr. P.P. Adab sir's lab in medicinal chemistry. Congratulations. Now, Durgesh Balodi. Five. And next winner is P. Sahu. Please come to the dais. Which lab you are from? Okay. He is from Dr. Kishore Mohan's sir's lab in medicinal chemistry. Okay. Congratulations. Okay. So Prasenjit is from Dr. AKT sir's lab, Amit Kumar Trivedi, in Cancer Biology Division. Thank you so much everyone for joining this quiz. I hope everyone have enjoyed the Little Brain Challenge. I would request, thank you so much ma'am. Yeah, I would request our guest of honor and director ma'am to please have their seat. So after enjoying this little brain challenge, since we are already in the spirit of solving puzzles, I would like to leave you with one final riddle before we move on. The more you chase me, the more I learn to escape. Every careless dose is a lesson. Every unfinished course an opportunity. I grow stronger with every mistake. So what am I? Yes, as we move into our next session, you will gradually uncover the answer for yourself and perhaps more importantly, discover why it continues to challenge the researchers, the clinicians, and healthcare systems around the world. With that, I would like to invite our next speaker, Sonam, to take us through the next session. Sonam, please. Thank you. वो कहाँ है?
Good afternoon, everyone. My name is Sonam, and I would like to start this presentation with a very simple question. What would modern medicine look like without antibiotics? Imagine that it is the year 1928, a simple cut on the skin, a minor wound, or a routine infection could easily become a death sentence. Doctors had very few options, and millions of people have died due to bacterial infections that today seem easily treatable. Until Alexander Fleming noticed something unusual growing in a petri dish. That observation led to the discovery of penicillin and changed medicine forever. For the first time, humanity gained a powerful tool to fight against bacterial infections. Over the next few decades, antibiotics revolutionized healthcare. But there was one problem. Bacteria never stopped evolving. The more we relied on antibiotics, the more bacteria had the opportunity to develop resistance. Today, we are facing antimicrobial resistance or AMR, one of the greatest global health threats of our time. To understand why new antibiotics are urgently needed, let's first take a look at the journey of antibiotics from their discovery to the current challenge of resistance. Discovery of penicillin opened what we now call the antibiotic discovery era. During 1920s to 40s, 10 new antibiotic classes were discovered, which was followed by the golden age of antibiotic discovery. During 1940s to 1970s, 20 new antibiotic classes were discovered. It was an extraordinary period of scientific progress. However, that success did not last forever. As we moved into 1980s and beyond, only 5 new antibiotic classes were discovered. Reason being, scientists have already found many of the easily discoverable antibiotic classes. Also, developing new antibiotics became increasingly expensive and scientifically challenging. Also, pharmaceutical companies have shifted their investment toward more profitable therapeutic areas. As a result, only a handful of truly new antibiotic classes were discovered. Meanwhile, bacteria were evolving. AMR does not develop because of a single factor. There are multiple So first we would see how does bacteria develop drug resistance. Naturally, a bacterial population contains resistant bacteria. When an antibiotic is added, those resistant ones survive and the susceptible ones are killed. These resistant bacteria can further multiply and transfer their resistant gene to another bacteria. According to WHO, 7.7 million people die every year, out of which 4.9 million deaths are due to AMR and 1.27 million deaths are associated with bacterial pathogens which are resistant to antibiotics. Also, a recent analysis which was published in The Lancet have estimated more than 39 million deaths from antibiotic-resistant infection between now and 2050. AMR does not develop because of a single factor. There are multiple human, environmental, and microbial factors working together. One of the major causes of AMR is overuse and misuse of antibiotics, followed by the incorrect prescription practices. Also, the use of antibiotics in agriculture and animal farming can promote resistance. Also, inadequate hygiene, poor sanitation can also lead to the spread of resistant bacteria across community and healthcare settings. The global spread of resistant bacteria is facilitated by international travel and environmental contamination. Also, the substandard quality of drugs may expose the resistant bacteria to the lower concentration of drug promoting resistance. In the end, bacteria evolve through genetic mutation, acquiring resistance. But not all bacteria pose same level of threat. To help guide research and drug development, WHO has identified bacterial pathogens which need urgent attention. On that basis, WHO has identified the categories critical, high, and medium priority group. The organism highlighted here is Neisseria gonorrhoeae, which is a high priority pathogen recognized by ICMR as well as WHO because it has developed resistance against fluoroquinolone and third generation cephalosporins. Now let's take a closer look at the organism itself. Neisseria gonorrhoeae is a gram-negative diplococcus which is highly adapted to its human host. Clinically, males are more likely to develop symptoms while females remain asymptomatic. Apart from urogenital gonorrhea, Neisseria gonorrhoeae can impact extragenital and can also cause neonatal infections. According to CDC, approximately 1.6 new million infections occur every year due to Neisseria gonorrhoeae infection. And the biggest challenge is it has developed resistance to almost all the antibiotics which are used to treat it, be it ciprofloxacin, azithromycin, cefixime, spectinomycin, and gentamicin. And now, ceftriaxone is the only reliable, effective empirical option to treat these infections, and it is an intramuscular injection. Which brings us to the key question. Could a single oral dose antibiotic provide cure rate close enough to ceftriaxone? The drug which we are going to talk about today is developed exactly to answer this question. On December 12, 2025, FDA have approved 2 oral therapies to treat uncomplicated urogenital gonorrhea, gepotidacin and zoliflodacin. Gepotidacin is a novel class of antibiotic which target the gyrase A subunit of DNA gyrase and the part C subunit of topoisomerase 4. It has a 2-dose oral regimen, 3 gram each 10 to 12 hours apart. It is approved for gonorrhea as well as uncomplicated UTIs. On the other hand, zoliflodacin is a first-in-class pyropyrimidine trione antibiotic which targets gyrase B subunit of DNA gyrase. And the USP, it is a single 3-gram oral dose, which is developed specifically to treat uncomplicated urogenital gonorrhea. Now, I would like to call my colleague Sukanya to explain the chemical journey of zoliflodacin. Thank you.
Hello. Thank you, Sonam, for providing the background and highlighting the need for zoliflodacin. So, good afternoon, everyone. My name is Sukanya. Today, I am going to present the fascinating chemical journey of zoliflodacin. The journey started with high throughput screening of approximately 250,000 compounds from Pharmacia compound collection, whereas several thousand antibacterial compounds were identified. Then these filter on the basis of selection criteria such as activity against both E. coli and S. aureus, amenability to medicinal chemistry, level of serum binding, lack of antifungal activity, high degree of purity, and adequate inventory. After the prioritization process, only 14 compounds were selected, and one of them was PNU 286607 as a lead candidate. This compound showed broad spectrum activity against both gram-positive and gram-negative bacteria, including resistant strain, and low frequency of spontaneous resistance. Then its mechanism of action were identified to analyze how PNU affects global biosynthesis pathway. So radio-labeled precursor assay was done, which is revealed that this compound inhibits DNA synthesis pathway. So due to low incorporation of radio-labeled thymidine. To identify its target, its resistant mutant were developed that help increase susceptibility to no-biosine and decrease susceptibility to norfloxacin, which is indicate that it inhibits same enzyme to no-biosine and norfloxacin, which is topoisomerase 2, mainly DNA gyrase, and topoisomerase 4. So sequencing mutant revealed that mutation in gyrase V. This was further confirmed by genetic complementation assay. So PNU 286607 bind to the novel site on gyrase V distinct from fluoroquinolones. After mechanism of action identified, they initially evaluated a racemic mixture PNU 286607. This compound showed promising antibacterial activity with minimum inhibitory concentration 1 microgram per ml against S. aureus and 4 to 8 microgram per ml against E. coli. To confirm its target, they performed enzyme inhibition assay and found that it inhibits E. coli DNA gyrase with half inhibitory concentration 9 micromolar. This finding confirmed that the racemic mixture was active against bacterial DNA gyrase and making it a promising lead for the further optimization. However, because PNU was a racemic mixture and containing two different enantiomers, so the next question was whether both enantiomers contributed equally or only one was responsible. To answer this question, the racemic mixture was resolved in multi-gram quantities of plus and minus enantiomers. The result was very clear that the plus enantiomers was clearly identified as inactive, with showing MIC greater than 128 microgram per ml against E. coli and S. aureus, and IC50 greater than 100 micromolar. In contrast, the negative enantiomers showed excellent antibacterial activity, with MIC 0.25 microgram per ml against S. aureus and 2 microgram per ml against E. coli, and much lower potent DNA gyrase IC50 2.4 micromolar. This clearly demonstrated that almost all antibacterial and DNA gyrase inhibition activity resided in negative enantiomers. Although negative enantiomers showed potent activity, but it lacked the property of an ideal drug. The main challenge was nitroaromatic group, which generally considered a medicinal chemistry liability because of concern related to safety and metabolism. So instead of increasing potency, they focused to design a safer scaffold while preserving DNA gyrase inhibition activity. So further structure activity relationship optimization was carried out, and they explored two different series. First is tetrahydronephridine. Although tetrahydronephridine showed good antibacterial activity, but they were difficult to synthesize due to needed highly substituted pyridine and complex chemistry. Therefore, they focused that second series, that is benzoxazole, which eventually led to the discovery of AZD0914. Now let's look how to optimize the benzoxazole ring, which ultimately led to the discovery of AZD0914. This optimization was carried out by Dr. Gregg and his team. After replacing nitro-containing scaffold to the benzoxazole scaffold, they initially synthesized a racemic compound, that is compound 1. This racemic compound showed good antibacterial activity with MIC 2.8 micromolar against S. aureus and 100 micromolar against E. coli, and potent DNA gyrase IC50 of 0.38 nanomolar. Since it was again a racemic mixture, so separate it into individual form and tested it individually. Among all these stereoisomers, the negative one showed potent activity with MIC 1.0 micromolar against S. aureus and 45 micromolar against E. coli, and potent DNA gyrase IC50 0.29 nanomolar. And the other stereoisomers showed significantly lower antibacterial activity. Therefore, they selected the negative one stereoisomers as a lead confirmation for the further optimization. After locking active stereoisomers, they next evaluate the role of fluorine, replacing it with chlorine. Although chlorine atom showed similar DNA gyrase inhibition activity, but it reduced the antibacterial activity. Therefore, the fluorine was retained and used for further optimization. Next, they optimize the benzoxazole ring by introducing several different analogs. Although many analogs were synthesized and evaluated, and most of analogs showed moderate activity. Among them, The oxazolidinone substituted analog showed best overall activity profile with potent DNA gyrase inhibition and excellent antibacterial activity. Therefore, they selected the oxazolidinone ring for the further optimization. After selecting oxazolidinone ring, they further optimized this part of the molecule. This time, they introduced several different analogs at the carbon adjacent to the ring oxygen and explored both mono-substituted and di-substituted analog, and also replaced oxygen to the nitrogen to understand its effect on activity. Among all these analogs, the compound 1T showed best oral activity profile with potent DNA gyrase IC50 0.39 micromolar and the excellent antibacterial activity 0.20 micromolar against S. aureus and 6.2 micromolar against E. coli, and also exhibited favorable solubility. After identifying compound 1T from SCR1, they performed the second round of SAR, and at this time they modified at the carbon adjacent to the ring nitrogen. These several analogs were synthesized and evaluated. Most of analogs showed good to moderate activity, but the compound 1T demonstrated overall best activity profile with potent DNA gyrase IC50 0.17 micromolar and excellent antibacterial activity 0.20 micromolar against S. aureus and 6.2 micromolar against E. coli, along with high solubility. After completing SAR study, they identified two lead compounds, 1T from SAR1 and 1U from SAR2. Both compounds showed similar potency with excellent DNA gyrase inhibition and identical antibacterial activity. So at this stage, neither compound was clearly superior based their potency alone. To differentiate these two lead compounds, they next evaluate their safety profile by performing in vitro cytotoxicity study. As shown in this table, both compounds were non-toxic toward red blood cells and other somatic cells. However, the key difference was observed bone marrow toxicity, as the value indicate. Since according to a better cytotoxicity profile, compound 1U being selected for the further preclinical study. Now let's look at the synthetic approach used to prepare a successful lead AZD0914. The synthesis starts from commercially available trifluorobenzaldehyde. First, the aldehyde group protected as an acetal, then the formyl group introduced onto the aromatic ring and converted into oxime. Chlorination of oxime followed by cyclization and form benzoxazole ring. Then the chiral amino alcohol attached to the benzoxazole ring introduce a oxazolidinone ring via CDI mediated carbonyl insertion. Then deprotection of this acetal morpholine containing side chain is attached, and finally condensation with barbituric acid generate a spiropyrimidine trione core. Subsequently, cyclization through 1,5-hydride shift followed by epimerization afford desired diastereomer. After purification by supercritical fluid chromatography, AZD09 is obtained. Now let's briefly discuss the key synthetic challenge encountered during the synthesis of AZD0914. The first challenge was introduce expensive chiral dimethyl morpholine. To reduce its cost and improve overall synthesis, it is introduced at the late stage through aromatic substitution reaction. The next and most important challenge was spiropyrimidine trione core. This was achieved by reacting intermediate with the barbituric acid followed by 1,5-hydride shift. And however, the reaction produced a mixture of diastereomer due to epimerization. So therefore, the desired active diastereomers was isolated with the help of supercritical fluid chromatography and giving desired product AZD0914. That's later named joliflodacin. Now finally, look at the structure-activity relationship. This is spiropyrimidine trione core. This is essential for macophore and responsible for DNA gyrase inhibition and topoisomerase inhibition. Morpholine ring controls the 3D conformation and locking the molecule in their favorable conformation and affects the antibacterial activity. Fluorine substituent, increasing lipophilicity and improving membrane permeability. Benzoxazole ring, reduce toxicity and help to improving metabolic stability. After introducing oxazolidinone ring, it's helped to improve antibacterial activity and solubility and avoiding bone marrow toxicity. So as you have seen that joliflodacin is initially screens against S. aureus and E. coli. Among them, it showed potent activity against S. aureus. which is gram-positive pathogens. Since discovering antibiotic that's effective against gram-negative pathogens is challenging, so joliflodacin against tested against other gram-negative pathogens, which will be discussed by my colleague. So for that, I would like to invite my colleague, Ms. Ritu, for further. Thank you.
Thank you, Sukanya, for taking us through the chemical journey of joliflodacin. Good afternoon, everyone. My name is Ritu Mandolia, and now I am going to discuss about the activity of joliflodacin against gram-negative pathogen, particularly Neisseria gonorrhoeae, and its preclinical development for urogenital gonorrhea. But before we dive into those studies, let's first understand how its primary target, DNA gyrase, operates within bacteria. So, in normal scenario, during DNA replication, the double-stranded DNA begins to unwind so that it can be copied. But as it unwinds, it creates twisting tension ahead of replication machinery. The twisting tension increases so much that the uncopied segment of DNA wrapped around itself to form supercoils. If this tension is not relieved, the replication eventually stops. This is where DNA gyrase comes into action to reveal the twisting tension generated during DNA replication. In the process, two segments of DNA binds to the enzyme, then enzyme cleaves one segment of the DNA, creating a transient cleaved complex. Another segment passes through the cleaved segment, then enzyme religates the cleaved segment. By doing this repeatedly, DNA gyrase introduces negative supercoiling and thus relieves the twisting tension generated during DNA replication. Since DNA gyrase is essential for bacterial survival, it serves as an excellent target for antibacterial drugs such as fluoroquinolones and as well as joliflodacin. In the presence of joliflodacin binds to the gyrase B subunit of the DNA gyrase, which stabilizes the cleaved complex. That prevents the religation of the cleaved segment. That leads to the accumulation of DNA breaks, causing replication fork collapse and ultimately bacterial cell die. Next, to identify the distinct binding mechanism of joliflodacin with DNA gyrase, several in vitro assays were performed, which showed that joliflodacin strongly stabilizes the cleaved complex at low micromolar concentration, at CC50 value of 1.7 micromolar, which is the concentration required to inhibit 50% of the cleaved complex. It also showed low activity towards human topoisomerases, indicating its bacterial target selectivity over human topoisomerases. In comparison to fluoroquinolones that require metal ion bridge for their binding to gyrase A subunit, joliflodacin binds directly to gyrase B subunit through hydrogen bonding. Through mutational studies, the key residues involved in the binding of joliflodacin to gyrase B subunit were found to be aspartate at 429, lysine at 450, and serine at 467 position. Thus, joliflodacin target DNA gyrase through a novel gyrase B binding site, which is distinct from fluoroquinolone. Next, to determine the antibacterial activity of joliflodacin against Neisseria gonorrhoeae isolate, antibiotic susceptibility assays were performed, in which joliflodacin showed high antibacterial activity against Neisseria gonorrhoeae isolate, with most isolate showed MIC value of 0.12 mg per liter, indicating that only a low concentration of drug is needed to inhibit the bacterial growth. This low MIC was consistent across all geographic regions. Also, among the drugs tested, joliflodacin showed the lowest MIC distribution, meaning it inhibits the most bacterial isolate at lowest concentration. Moreover, joliflodacin showed equipotent activity against diverse and reference strains of Neisseria gonorrhoeae, including fluoroquinolone resistant and XDR, which is extremely drug resistant strains of Neisseria gonorrhoeae. Next, to investigate how easily bacteria develops resistance to joliflodacin, spontaneous mutation assays were performed, in which the first step mutants emerges at very low frequency, with the initial mutations emerges in gyrase B gene at aspartate 429 and lysine 450th position, resulting 16 to 32 fold increase in joliflodacin MIC. However, the second step mutants emerges only after extended incubation, with additional mutations emerges in gyrase B gene at serine 467 positions, resulting extra 2 to fold increase in jolly fluorescent MIC. Also, these mutants showed no cross-resistance with the standard antibiotics. Thus, jolly fluorescent maintains potent activity against global and drug-resistant nitroguanidine isolate while exhibiting a low propensity for resistant development. Next, to determine whether jolly fluorescent showed bactericidal or bacteriostatic effect, or whether the effect is concentration dependent or time dependent, time-kill kinetic experiments were done. In the susceptible Neisseria gonorrhea strain, when the concentration increases from 1x to the 8x or 16x MIC, a rapid reduction within few hours in bacterial count was observed. Thus, jolly fluorescent showed rapid and concentration-dependent bactericidal activity. This potent activity is also retained against XGR isolate. Thus, jolly fluorescent acts as a rapid concentration-dependent bactericidal agent. Then to know how jolly fluorescent was absorbed, distributed, metabolized and excreted from the body, different animal models were used, in which after oral dosing, jolly fluorescent was rapidly absorbed with the peak plasma concentration reached within 1.6 to 3.1 hour. Jolly fluorescent also showed moderate to high oral bioavailability. Moreover, jolly fluorescent shows moderate plasma protein binding across all species. Also, jolly fluorescent is mainly metabolized by phase 1 and phase 2 pathways. However, low potential for major CYP-mediated drug-drug interactions were seen. It is mainly excreted through hepatobiliary or fecal pathway, with small proportion contributes to urinary excretion. Therefore, jolly fluorescent demonstrated favorable ADMA characteristics supporting oral administration. Next, before we understand the in vivo efficacy of jolly fluorescent, let's first understand how the PKPD of antibiotics is determined. So, in case of antibiotics, MIC shows how susceptible the bacteria is, but MIC alone is not enough to decide the dose. So, that's why we need PK, because in the body, drug level changes with time. So, combining PK with MIC gives the optimal dose and dosing strategy. And based on how different antibiotics kill bacteria, there are three major PKPD indices. First one is the time above MIC. It tells how long drug level stays above MIC. Antibiotics which exhibit time-dependent killing, such as beta-lactams, follow this PKPD index. For these antibiotics, the main goal is to keep the drug level above the MIC. So they are given as more frequent doses or by prolonged infusion. Second one is the Cmax over MIC. It tells how high the peak is relative to MIC. Antibiotics that exhibit concentration-dependent killing, such as aminoglycosides, follow this PKPD index. For these antibiotics, higher concentration peaks gives better killing. So they are often given as larger single or less frequent doses. Third one is the AUC over MIC, which tells total drug exposure over 24 hours relative to MIC. Antibiotics that exhibit exposure-based killing, such as fluoroquinolones and vancomycin, follow this PKPD index. For these antibiotics, both the concentration and the duration of the exposure contributes to the bacterial killing. So their total daily exposure is optimized either by adjusting dose or dosing intervals. Now, if we talk about jolly fluorescent, hollow fiber infection model was used to identify the main PKPD driver of jolly fluorescent efficacy. It is an in vitro developed model that simulates human body, in which drugs were given as once daily, every 12 hours and every 6 hours. Among all the PKPD indices, AUC by MIC shows the best agreement between the observed model and predicted model, with the highest R-square value of 0.95, which indicating a strong correlation with the bacterial killing. And it also has the lowest WSSR of 42, indicating least prediction error. Thus, AUC over MIC is identified as the primary PKPD driver of jolly fluorescent efficacy. The next objective was to determine the exact PKPD value required for effective bacterial killing. For that, mouse thigh infection model was used. Since Neisseria gonorrhoeae is the human-specific pathogen, S. aureus was used as surrogate pathogen to stabilize the PKPD target, in which two important PKPD targets were determined: the bacterial stasis target and bacterial killing target, in which showed that to achieve the bacterial stasis, the free drug exposure relative to MIC should be 66, meaning to achieve the bacterial stasis, the free drug exposure should be 66 times higher than the pathogen MIC. And to achieve the bacterial kill, the ratio should be 139. This SODS PKPD target was translated to Neisseria Gunedi PKPD target. Then PTA analysis was completed. It is the probability of target attainment. It is done by the virtual clinical trial to identify the efficacious dose that covers the PKPD target in more than 90% of the subjects using Monte Carlo simulation. This analysis showed that a dose of at least 800 mg would be required to achieve the bacterial stasis target and at least 1600 mg to achieve the bacterial kill target. These PKPD studies provided the scientific basis for selecting the clinical doses. Then to identify the safety profile of joliflodacin toxicology studies in rat and dog for 1 month was conducted. The adverse effect was in which the adverse effect such as irregular respiration, hematopoiesis, infertility, testicular degeneration, reduced pregnancy was seen at 500 and 1000 mg per kg per day dose in rat, and testicular degeneration and tachycardia at 200 and 500 mg per kg per day dose in dogs. However, most of these adverse effects were reversible after few months, and a NOAEL of 200 mg per kg per day dose in rat and 100 in dog was established. The NOAEL exposure was approximately 13 times higher than the efficacious exposure observed in mouse thigh infection model. Thus, joliflodacin produced effective antibacterial killing at exposures far below those associated with the toxicity, providing a 13-fold safety margin. The novel mechanism and antibacterial activity of joliflodacin along with its safety profile supported the IND filing and progression into human clinical trials. Now, I would like to invite Sonam to discuss the preclinical trials. Thank you.
After the successful completion of clinical studies, zoliflodacin entered into the phases of clinical trials. As discussed in the previous session, for oncology drugs, phase 1 clinical trial is conducted in patients. On the other hand, for non-oncology drugs, phase 1 clinical trial is conducted in healthy volunteers. Now, let's take a closer look at the journey of zoliflodacin through the phases of clinical trial. Phase 1 clinical trial was conducted in healthy volunteers to ask 3 key questions. Is the drug safe? How well is the drug tolerated? And how does the drug behave in the body after administration? To answer these 3 key questions, a random placebo-controlled open-label study was designed. This study had 2 parts, single ascending dose study and mass balance or ADMA study. In the first study, participants have received increasing dose of zoliflodacin from 200 mg up to 4000 mg. It also included a food effect study. The second study focused on the pharmacokinetic properties of zoliflodacin, and the results were very promising. Zoliflodacin was very well tolerated up to 4000 mg with no serious adverse events reported. Also, it has achieved the maximum plasma concentration in 1.5 to 2.5 hours in case of fasted state versus in around 2.5 to 4 hours in case of fed state. Also, zoliflodacin have showed nearly dose proportional PK up to 3 grams, and food have delayed the absorption of the drug, and it was extensively metabolized from the body before excretion through the feces route, which was the major elimination pathway. Also, 3 gram have provided the optimal pharmacokinetic parameters which were required without the need of increasing any higher dose. So, phase 1 trial have successfully established that single oral dose up to 3 grams zoliflodacin have a favorable safety profile and exhibited linear pharmacokinetics. There were no serious adverse events were reported during this trial. After the findings, zoliflodacin entered into the phase of clinical trial 2, phase 2. The objective was to evaluate the efficacy and safety of zoliflodacin as compared to the standard of care ceftriaxone in male and female subjects with uncomplicated gonorrhea. It was a multi-center, random and open-label study. Patients with positive gonorrhea diagnosis who were male or non-pregnant female were included for this study. Also, these participants have been divided into 3 groups. The first group have received single oral dose of 2000 mg zoliflodacin. Group 2 have received single oral dose of 3000 mg zoliflodacin, and Group 3, which was the active comparator group, have received 500 mg intramuscular dose of ceftriaxone. And the endpoints were to evaluate the microbiological cure of urogenital gonorrhea at rectal, also with rectal and pharyngeal infection sites. Also, zoliflodacin was evaluated for its antimicrobial activity. If we talk about the efficacy results, zoliflodacin have shown comparable efficacy to ceftriaxone and shown 96% cure rate to treat urogenital gonorrhea. And in case of rectal gonorrhea, zoliflodacin have performed extremely well and shown 100% cure rate as compared to the standard of cure. In case of pharyngeal gonorrhea, the 3-gram dose of zoliflodacin have shown higher efficacy as compared to the 2-gram dose of zoliflodacin, and there were no serious adverse events were reported during this trial. Also, zoliflodacin was assessed for its antimicrobial activity where against the gonococcal isolates and compared with the known antibiotics like azithromycin and ceftriaxone. Zoliflodacin was found to be 100% susceptible against all the gonococcal isolates. From phase 2 clinical trial, 3-gram dose was selected for further clinical development. Now let's move towards the phase 3 clinical trial. It was a multinational trial which included 17 sites across 5 countries: USA, Belgium, Netherlands, South Africa, and Thailand. The objective of phase 3 trial was to demonstrate the non-inferiority of a single 3-gram oral dose of zoliflodacin versus standard of cure 500 mg single intramuscular dose of ceftriaxone plus 1 gram single oral dose of azithromycin. The original article was published in the Lancet. It was an international, random, open-label and non-inferior study. And non-inferiority trial shows that a new drug is not meaningfully worse than the standard of care. And for zoliflodacin, the non-inferiority margin was less than 12%. Now, let's talk about the study design. Participants who were 12 years or older, having suspected uncomplicated gonorrhea and were willing to provide their informed consent were included in the study. These participants were randomized in a 2:1 ratio to receive either zoliflodacin or the comparator. Around 620 patients have received 3-gram single oral dose of zoliflodacin, while 310 patients have received ceftriaxone 500 mg intramuscular dose plus 1 gram oral azithromycin. After the treatment, the patients have been called for test of cure visit, which is a follow-up visit to check whether the infection have been completely eradicated or not after the treatment. For zoliflodacin, it was day 6 plus minus a window of 2 days. And the endpoints were to evaluate the microbiologic cure rate at urogenital, pharyngeal, and rectal sites in micro-ITT population. Also, zoliflodacin was assessed for adverse events and safety evaluation. Now, let's talk about the results from the study. First, they have assessed the microbiologic cure rate at test of cure visit at urogenital site in micro-ITT population. Micro-ITT population includes actual infected patients who had a confirmed gonorrhea infection before treatment. In this population, zoliflodacin have shown around 91% cure rate as compared to the standard of cure, and it remained within the non-inferiority margin. The next analysis was evaluables or sensitivity analysis, which included only those participants who have completed the study according to the protocol and returned for the test of cure window visit within the required time window. In this case, the findings were consistent and zoliflodacin remained within non-inferiority margin. Not always the patient returned in a given specified time window. The next analysis included all the patients with confirmed infection, even if they came for the test of cure visit outside the scheduled time window. Zoliflodacin performed very well and shown around 93.1% cure rate in case of this population as compared to the standard of cure. The next analysis was micro-ITT excluding key protocol deviations, which included only patients with confirmed infection who have followed the study protocol properly and In this population, zoliflodacin have shown around 91% cure rate as compared to the standard of cure, which has shown 96.2% cure rate. And among all the analysis, zoliflodacin have remained within the non-inferiority margin. Now, after the urogenital sites, researchers also assessed the microbiological cure rate at pharyngeal and rectal sites. In case of pharyngeal site, in micro-ITT population, zoliflodacin have shown cure rates around 79.2% as compared to the standard of cure. In case of evaluables, zoliflodacin performed very well and shown around 91.3% cure rate as compared to the standard of cure. It also included per protocol population, who are the participants with confirmed gonorrhea who completed the study according to the protocol and received the assigned treatment correctly. And also they have attended the test of cure visit within the scheduled time window. In case of rectal gonorrhea, the findings were consistent and reliable. So the key highlights from the phase 3 clinical trial: it has shown higher cure rates for urogenital, pharyngeal, and rectal gonorrhea. Also, the findings were very consistent with the phase 2 clinical trial, which supports zoliflodacin as a promising oral alternative to treat urogenital gonorrhea. So, the phase 3 clinical trial established 3 gram single oral dose of zoliflodacin as a non-inferior alternative to ceftriaxone-based therapy for uncomplicated urogenital gonorrhea. And finally, after the completion of phases of clinical trial, Innoviva Specialty Therapeutics and GARDP have filed for new drug application, which was accepted in June 2025. And on December 12, 2025, FDA have approved zoliflodacin to treat uncomplicated urogenital gonorrhea. It is marketed as nasal vents. And the administration of zoliflodacin is very simple. You take 3 grams of zoliflodacin, mix it into 60 ml of water, and you are good to go. If we take a glance at the clinical journey of zoliflodacin, it took almost 13 years to develop this antibiotic. Phase 1 clinical trial started in 2013, which was sponsored by AstraZeneca and Intesys Therapeutics, followed by Phase 2 trial, and then Phase 3 trial was sponsored by GARDP and Intesys Therapeutics, which was later on acquired by Innoviva Specialty Therapeutics, who have filed for the NDA application for zoliflodacin. Also, FDA have approved zoliflodacin for the treatment of uncomplicated urogenital gonorrhea on December 12, 2025. After the approval of zoliflodacin, it will be continued to monitor against the resistance development, its long-term safety, and real-world effectiveness studies in the patients. Also, additional clinical research will help us understand the drug better. To conclude, I would like to highlight the mechanisms of the key points of zoliflodacin. It has a novel mechanism of action. It is an oral single-dose antibiotic and shown phase 3 proven non-inferiority to ceftriaxone-based therapy. Also, it has found to be active against MDR strains. And finally, zoliflodacin is surely going to have a strong global impact. Thank you.
Thank you all. If anyone has questions.
Thank you very much. So I from the last slide, I want to ask one question that you said it is a single dose, right?
Yes, sir.
Now, as far as remember, the T-half is something around 2 hours.
Yes, sir. 2.5 hours.
2.5 hours. So if you think about the total half-life of the molecule, so that means by 7 hours, the compound will be completely excreted from your body.
Yes, sir.
So then within 7 hours, compound is excreted from your body, and then still a single dose is effective. Would you like to, put some light on that?
It has been calculated for both fed and fasted state. It can be excreted, but I don't have actual explanation.
And one more question from the first slide, that in the inclusion criteria, what you have mentioned that the lack of antifungal activity. So what was that? So just to minimize the false positive data from the antifungal activity?
Yes, sir.
Oh. Thank you.
Yes, sir. It was completed in 2015.
Yeah,
Yeah.
2015. But approval took 10 years. Why? I mean, is there any mention or something?
Yeah, it started from 20.
Phase 1, 13 started.
Phase 1 started in 2013.
When did the phase 3 end? Phase 3 end?
Phase 3 ended in yeah.
When? In
2015. 25.
25. 13 में जब start.
Yeah.
हो सकता है typo.
Yeah. Okay. 25.
14 का आपको लिखा है ना? No, इसमें incorrect.
No, I think it's 2015. Maybe.
13 में पुरानी slides में जाता हूँ. Go to the previous slide.
Previous slides.
Go to previous slide. It's wrong. There is some. नहीं सही कह रहे हो.
Sir, I think it's a typo.
It may be a typo. पुराने में आपने किस 3 से start करें? हमने जो है जगह.
Sir, this is the paper actually.
Lancet 26.
The paper was published in 2025.
Probably typo. Yeah, probably typo.
It's a typo.
That's what I was wondering.
I just comment on that. One of the problems with antibiotics is that there is very little investment, right? So if you look at the number of hands that this particular molecule changed, it went from Astra to Entasis to Inovia, and finally it was GARDP that came in and funded the phase 3. And the reason for this is that even though everyone agrees that we need antibiotics as a public good, there isn't enough investment. That has caused this huge delay in the clinical development of this compound. Think about the Protac one, which started clinical development in 2019, was approved in 2025. This one started clinical development in 2013 and was approved finally in 2025. It just reflects the differences in investment in the two areas. And Dipankar, going back to your question, it is 3 grams. The PK study was done at whatever lower dose it was. So 3 grams, even with that half-life, as you keep on eliminating from the body, you're still achieving AUC levels which are sufficient to drive efficacy. So the AUC by MIC ratio, which she was showing, they are able to sustain that over a large part of the 24-hour period. And that is why it works as a single dose. Yeah, but it depends on what you're starting with, right? It's AUC. 3 grams you're starting with. So as you, even as you eliminate. Yeah, okay. Any other questions? Otherwise, let's move to the next phase.
Not included.
I think, sir.
But any some reason behind it? Or?
Yeah, sir. There were only patients who were having very highly cases of these kind of infections.
So that India is lacking that.
Yes, sir.
So it's a good news.
Maybe
so
like,
it's a good news. Because you showed that because there is no Indian.
yes, sir. There were only 5 countries which were
only
involved.
5 countries.
Yes, sir.
Oh, that's great.
Yes.
Oh.
So after traveling through the exciting journey of epidegistran and zoliflodacin, now I would like to introduce our chief guest, Dr. Swami Subramaniam, Director, Ignite Life Science Foundations. It's our honor and privilege to welcome our esteemed chief guest, Dr. Swami Subramaniam, a distinguished physician, scientist, visionary leader, and innovator. Trained as a clinical pharmacologist from PGI Chandigarh and molecular neuropharmacologist from the University of Pennsylvania, Dr. Swami has contributed significantly to both academic research and pharmaceutical innovation. With over 2 decades of leadership experience, he has driven landmark initiatives, including India's first IND filing for novel anti-cancer drug, founded Origin Discovery, and held key positions at Dr. Reddy's, Merck & Company, and Abbott Nutrition. A recipient of prestigious Gurukul Chevening Fellowship, he has also trained at Stanford, IIM Ahmedabad, and Indian School of Business. Beyond his scientific achievements, he is the author of Mastering Sleeps and Healing Hand. We are truly delighted to welcome Dr. Swami and look forward to his valuable insights.
Thank you, everyone, for staying so late after those really interesting presentations. Thank you to the director, Dr. Radha Rangarajan, for inviting me. I have to confess that the moment she invited me, I accepted immediately. This is not something I would want to miss. And it's truly been a good experience for me. It's the last yesterday meeting some of the senior scientists and today attending these 2 presentations. So now I was thinking about what I should speak about. When I saw the program, there was 10 minutes given to me and I said, what can I speak about to this audience? Many of you, the senior people, are experts in drug discovery and drug development. I can't tell you anything more than you already know. And the students, I realize that this is targeting students. So I have actually made my presentation much more aligned towards what students might be looking for. Will this work? That's the pointer. I put my pocket. Okay. Yeah. Okay. So I just thought I'll take the 10 minutes to cover 12 points. Right? There is science. Science is the production of knowledge, the creation of facts, and there is the science of science, which we don't think or talk about, at least in a conscious way. That is, how do we do science? If you look, and we don't study the process of doing science as much as doing science itself. So as a result, a lot of science is done like cottage industry. We still do it like it's a small shed in which a carpenter is making furniture. It is not sufficiently exposed to the processes and theories and practices that are, for example, widely prevalent in the manufacturing industry. Now, you may react to that saying this is not manufacturing, it has to be treated differently. And I think I would dispute that at least at several levels. I think the question of efficiency of doing something, getting everything right the first time, quality by design, right? Six Sigma. These things apply as much to science as to anything else. But we hardly give any thought to it. And I think this student audience, who many of you will become scientists in the future, perhaps you would benefit, at least in your careers, thinking about the science of doing science and thereby doing better science, right? So I have listed these 12 things here, but I've intentionally, in the quiz format, right, left out the second part of what I'm going to talk about. So I would leave it to you to guess how you complete each of those things. But let me start with the first one. So point number 1, right? And I have to take help of my notes here. So science is like, right? That is the first one. So can you guess what is science like? And again, this is idiosyncratic. This is not a standard textbook answer. But science, in my opinion, resembles two games, right? One is Lego and the other is Jenga. Let's take Lego first. What is Lego? Lego consists of blocks, bricks that you attach to each other. Science has that nature. Your science does not stand alone. It is interdigitating with the science other people do. And you have to be consciously aware of that. The reason you have to be consciously aware is, one is, your science has to be informed by what other people do, and not just people who have done it before you, but people who are doing it next to you, for example, in adjacent areas of science. We had a meeting in Bengaluru on epithelial mesenchymal transitions, which is a phenomenon studied extensively in cancer. But EMT is also seen in wound healing. It is also seen in development biology. But if you ask the development biologists, have they ever spoken to an cancer biologist about what they are studying? Most often the answer is no, they have not. And I think that is a failure of the science of science. We should actually be systematically talking to other people about our topic from their perspective, right? So in this meeting, we brought together the wound healing people, the people who study development biology, and the people who do molecular oncology. So I think the interfaces between those disciplines create new ideas for doing research. So that's what I meant by the Lego concept. What is Jenga? Jenga is the game in which you place blocks one on top of the other and you try to build a tall thing, and then the other person has to remove something from the bottom without toppling it, right? The point here is that the knowledge we create today is like a block of Jenga. The knowledge we create, somebody else creates in the future, is another block of Jenga on the top. Now, if the blocks of Jenga on the bottom are not sturdy, then you are creating an edifice that is not going to grow very tall. So I think we have to realize that we are not alone in this game. Everything we do connects with what other people do currently on the sides and with what people will do after us in the future, because they are going to reread our paper and say, believe it, and base their design and their experiment based on that. So it's a responsibility we have unlike in many other situations, scientists today are building the science for tomorrow, and we have to realize that, and therefore there is need for robustness in science. The other thing, I mean, in terms of the Jenga analogy, Sidney Brenner discovered messenger RNA in 1960s, right? We had the COVID vaccine, mRNA vaccine in the 2020s. Now, if Sidney Brenner had screwed up, or somebody along the way had screwed up, the mRNA vaccine could have been delayed at least by a few years, right? So imagine the consequences of screwing up on your science when you do it. The next point is design defeats. What does design defeat, right? Design defeats uninterpretability. Many of the experiments we do gives us data that we cannot interpret. The reason is we have not designed the experiment properly. We need the right controls, positive controls, negative controls. We need the right checks. Now, I'll give an example again here. We funded a project where a drug was to be tested in vivo after having passed in vitro testing. So what is the first thing you would do when you want to do an in vivo testing of a drug that seems to work in vitro? You would see whether it is stable in plasma, right? You would see whether there is adequate bioavailability when you give it in vivo. So the scientists went ahead and did it in vivo, and it had no effect. So then they analyzed the plasma. There was no drug in the plasma. It turns out the drug had an ester bond, and plasma esterases were cleaving it. So they should have done the simple task of checking for plasma stability before going from in vitro to in vivo, which is a very simple thing, but we don't think about it because we are so married to our in vitro biology that we don't realize that the transition going from in vitro to in vivo is not as simple or straightforward. Choosing the right dose, the right route of administration, making sure the compound is stable, has sufficient exposure that we think we need based on our in vitro experiments are critical things we need to do. So these kinds of design issues can defeat uninterpretability. Otherwise, you will get data that you will not make sense of. The next is data defeats theory. So there are lots of theories, and for example, let's take homeopathy, right? That if you ask homeopaths, there's a theory for homeopathy. It's not as if they are just doing it out of air. They say there is these molecules make some changes to the other molecules of water, and that then causes a beneficial effect. But the data, randomized clinical trials have shown that homeopathy does not work. So ultimately, data has to inform theory. While theory can be used to design the experiment to do it, but eventually data has to win at the end of the day. Point number 4. This is in German because, again, there is an anecdote associated with it. Ein Versuch is kein Versuch. I think some of you who know German or have some familiarity or may have even read this before will understand immediately what it means. Ein Versuch is one trial, is not a trial. That's what it's in German. What happened was we had visiting scientists in the lab at NIH from St. Petersburg in Russia, and he was leaving the lab, and he gave me a German to English dictionary as a gift, parting gift. I don't know why he chose to give it to me, because I was not particularly learning German or anything at the time, but maybe he wanted to give away all the books in his attic, and he gave it to me. But he wrote on the top of it, Ein Versuch ist kein Versuch. And maybe he wrote on it with the intention that I will read it and then try to find out what it means in the dictionary. So maybe, so that I think that's how I came across that thing. But this is a very important thing, right? So I remember another anecdote is, and this has a connection with CDRI, I believe. This is Brahmi. Brahmi was being developed here for cognitive. There's a product called Mentat. It was on the market as well, right? I was at the NIH working on hippocampal neurons, studying primary cultures, doing electrophysiology. So I was recording channels in these cells. And I said, it'll be, it's a crazy idea, bad design. I did an uninterpretable experiment. I sourced Brahmi extract from India. I think it was from somebody from CDRI who sent it to me, and I started, Arvind would get very upset that I did that. I used the system to put some Brahmi extract on the cells, and of course they started going all kind of excitation. I also got very happy. I said, oh, wow, seems to be excitatory. Maybe that's how it is improving cognition, etc. And I went home very happy. And the next day I came and repeated the experiment, didn't work. So Ein Versuch is kein Versuch is what I learned from that. Then the next point is the failed experiment, right? I think we are very happy. We have a hypothesis. We do an experiment. We are very happy when the hypothesis is proven, because then we can quickly go into publication, right? We can publish a paper. But I actually think the most interesting results are the ones where the hypothesis is disproven, because that is showing you new biology which even you had not anticipated. So every experiment where your hypothesis, which you or your pet hypothesis, which you want to prove, is disproven, is the most interesting experiment. That is where you will get really interesting results that other people will respect. You will get a paper from the first one by proving a hypothesis that everybody else has. You will get a paper very fast. But the interesting result is the outlier data. So look at your data with the outliers in your data, especially if it is consistently there. You know that the experiment is trying to tell you something. It is talking to you. Listen to it and try to find out what that outlier actually means. And you might come up with something new. The next is quality of tools. What I meant by this is we have come to an age where we are increasingly increasing the instrumentation that we have at our command, right? So we are using a lot of sophisticated instruments to cryo-electron microscopy. We are doing things that we could not have conceived of even when I was in industry, right? The amount of high throughput things we do, even the CSIR assay. We never had the we were not doing CSIR assays to identify targets when I was a industrial researcher. So we have a lot of tools in our hands. The quality of the tools is a big differentiator. So unless you have high quality tools, even things like cell and culture, unless you have good healthy cells and culture, you're not going to get meaningful results. So we have to pay a lot of attention to the quality of tools in our disposal. So and one example comes to me. Fleming is actually an example of a failed experiment. In terms of the quality of tools, we are also having more refined and more sensitive instruments now. We are detecting phenomena that we could not have detected 20, 30 years back. The next is telescope vision. Yeah. So in science, we take a slice vision. That is, we do an experiment, we publish it, and we forget about it. I think it is important, especially in a place like CDRI, we develop telescope vision. We say, what next? What next? We don't say it after the experiment is done, well before the experiment is even designed. We say, okay, if I get this result, what would be next? And that's why we actually encouraged some of the people we funded to develop a target product profile, because it actually starts with the end goal in mind. You have a target product profile, and then you work back towards the experiments you're doing that will discipline you in telling you what the experiments you should do are. There are some nice to-dos which might be in the periphery, orthogonal to the main thing. But there are always a critical set of experiments that would take you fastest to the target. And unless you do that kind of exercise, you might end up just meandering through many side alleyways, which are just convenient because those reagents are available in your lab. You just spend a lot of time doing that, which is actually not critical to achieving your goal. So you have slice vision, slice vision is basically a vision of what you do and what the other people in your field are doing on that day. But also think about the future, telescope vision. The next is self-criticism. I think you have to be the biggest critic of your own data. So we review a lot of projects, and we ask them the questions they should have actually asked themselves before they came to the review meeting, right? So we ask them a question, they'll respond at that point of time in a reactive manner. But I think we all should look at our data and say, what is wrong with this data? Can I critique this data? Can I explain what it is? Or if there is some defect in it, can I fill that gap before I go into a review meeting, right? So I think we should be the biggest critics of our own data. I think there's a lot of value in doing that. Then the next one is, you think you do this, but I'm sure you don't do enough of it. This is thinking. So we don't spend enough time thinking. So we spend a lot of time doing because we think doing is considered the highest value thing. But I think in science, thinking is much more important than doing, because if you don't think through things carefully, then you will do the wrong things which are completely useless, right? So I think we should spend, if you spend 1 hour doing experiment, you should spend 10 hours thinking about that experiment before you actually do the experiment. That is the kind of ratio. And thinking 24/7, you might say, how can you think 24/7? We need to sleep. And I just want to point out here that even when we sleep, our brain is active. And there is this story about Ramanujan, that he would get some of his best mathematical ideas in his sleep. And the reason he was getting his best mathematical ideas in his sleep was not because his brain while sleeping was somehow different. It is because he spent the rest of the day before falling asleep thinking about the mathematical problem. And the subconscious brain has a way of reflecting on things you've thought the previous day and identifying solutions. So thinking is very important, especially in this day of social media and internet and so much information being loaded on us. Okay. Next, and I think this is the penultimate, so we'll be done soon, is AI. And I don't have very good, I think there are people here, Atul, and people who are already using AI in your work. And I don't know how this is going to develop. I'm not an AI expert, but I think we have to be very much aware of it, and we need to evolve our science and research along the using the AI tools and co-evolve along with AI. I mean, that's the level of my expertise. I can't say more than that. The other thing is, one of the resources we have at our command is time, and we use it very inefficiently. For example, institutionally, for example, if I would say that if funds are delayed by 1 month or 2 months, actually it is delayed sometimes much longer than that. It is as good as telling the scientists, put your brain on ice. You don't need to use it, right? And then come back. And our scientists are the biggest value creators. We may be paying them not very high, but in terms of the value they create, it is the greatest value is created by scientists. And for us to delay by 2 months or 3 months funding so that the scientist is not able to get on with their work is a massive waste of a resource, which we don't realize. If we put a dollar value or a rupee value on how much economic value is lost because we have delayed funding or delayed decision making, or even at an individual level, you delay doing an experiment or delay doing something that's needed to be done, that's a huge economic loss. And we need to start putting value on that, which is much bigger than anything else, I think. This is really massive. The other thing is, of course, if you don't do things on time, you're going to be scooped by the Chinese, or somebody is going to file a patent that you should have been filing, right? So time is a very important resource, because in academia, at least, you tend to take things a little light. I don't know how it is in an institution like CDRI, but I know that universities, this is the academic mentality historically has been. I mean, I'm not saying that we should not have a little bit of time for ourselves and things like that. We should have family time and all those things. But even when you have family time, I think thinking, don't have to stop thinking, and thinking itself a way of productively using those downtimes. Finally, we all are, I mean, many of you are, and many of the students will be doing this. You are creating the knowledge and tools that are going to be useful tomorrow. So it is your personal responsibility to make sure you're doing the best of it. So I'll stop there, and I don't think there'll be any questions, but I'm happy to engage on if anybody has anything to say. There is a TEDx talk I gave on creativity, which is on YouTube. So you can search by my name and you'll find it. That's where I talk about Ramanujan, because it's interesting how Ramanujan comes up with his insights, which even people like his mentor Hardy and Littlewood they just cannot explain how this person does it. And I have attempted an explanation for you for Ramanujan's genius. And one of the main things I said is, it's very simple. You think 24/7 about your science, you will become a Ramanujan. Right? So thank you very much, and I wish you all the best. Assume there are no questions.
So, Mike.
Now I would like to request Director Ma'am, mentees Dr. Deepak Datta, Dr. P.P. Yadav, and Dr. Arnav Das Gupta to felicitate our chief guest.
Excellent.
Okay. Now I would like for the presenters to come to receive their token of appreciation. Nitika. Soumya Ranjan. Sukanya. Ritu. Sonam. Lastly, myself, Udita. Yeah. Now I would like Director Ma'am and Swami Sir to give token of appreciation to our mentees. Yeah, mentors.
We are menting. After your talk, we are menting. After all your talk, we are menting.
Dr. Deepak Datta. Dr. P.P. Yadav. And Dr. Arnav Das Gupta. As we mark the successful completion of this seminar, I take this opportunity to express my heartfelt gratitude to everyone who contributed in making this event a success. First, I extend my sincere thanks to our Director Ma'am for her constant encouragement, support, and guidance. I would also like to express my deepest gratitude to our esteemed chief guest for joining us today and sharing his valuable insights. I would like to thank students of Hygia College, Aryavat College, Amity University, and Lucknow University Pharmacy Department for gracing this event. My sincere appreciation goes to all our mentors for their continuous guidance and support in organizing and successfully conducting this seminar. A special note of thanks to Ravindranath Sir, Kazim Sir, along with audiovisual team Sumit Sir for their dedicated technical support. Dr. Santosh Tukla for curating the online platform for conducting the quiz. Cafeteria team for their excellent arrangement and warm hospitality. Lastly, I thank the organizing team and all the participants for their active involvement and enthusiasm, which truly made this seminar meaningful and memorable. Thank you everyone for your presence and contribution. Thank you so much. Yeah. Yeah. Then with all the audience. Yeah.
All of you who have joined from other pharmacy colleges and university, please stay back. There is some refreshment after this closing session for you.
Thank you. Thank you. Very well. Very well done. Excellent. Presentations. Nice. Picture. Thank you, sir.
Thank you.
Thank you. Thank you. स्विच हो जाता ना वो दिक्कत क्या है नए सिस्टम पे मतलब सका मतलब नया खुल जाता मतलब हाँ हाँ प्रॉब्लम है आ जाओ जल्दी से